I'm Rebecca Inman, and I chose prompt B, the Meselson and Stahl DNA replication experiment. At the time, there were 3 standing hypotheses on how DNA replicates in reference to the original DNA strand, which serves as a template for replication. These 3 replication types are conservative, semi-conservative, and dispersive. In conservative replication, the original DNA strands are kept intact and paired, while strands produced by duplication are paired together. After the second round of replication, the original strands are still together, while each pair of replicated strands are paired together. Semi-conservative replication pairs an original strand with a replicated one. Thus, after 2 rounds of replication, the original strands are each paired with a duplicated one, while the 2 other helices contain none of the original DNA material, but are composed entirely of replicated material. For dispersive replication, the DNA material is randomly repaired together after replication. Statistically speaking, after 1 round of replication, this would result in 2 DNA helices, each with half the original DNA material and half material that was duplicated. The second round results in all 4 DNA strands with mixed materials. Initially, Meselson and Stahl grew E. coli in medium that had N15, a heavier and rarer form of nitrogen, as its only nitrogen source. Once all the bacteria had incorporated this heavy nitrogen into the nitrogenous bases in their DNA, a sample of that bacteria was taken. The remaining bacteria was placed in a medium with only N14, the regular nitrogen, and allowed to replicate. Again, after the bacteria had incorporated the lighter nitrogen into their DNA, a sample was taken and the remainder was placed into more N14 medium. Each time a sample was taken, Meselson and Stahl determined the presence of heavy, intermediate weight, or light bacterial DNA through equilibrium density gradient centrifugation. To achieve accurate results, the sample tubes were spun at high speeds for several days. This process resulted in separated bands of DNA fragments suspended in the solution, with the heavy DNA at the bottom of the tube, the light DNA at the top, and an intermediate weight DNA in the middle. The intermediate band represents DNA that contained both heavy and light nitrogen. After 2 rounds of replication, the tubes lacked a heavy band, but contained an intermediate band and a light band. Since there was no original heavy DNA left, this ruled out conservative replication, but the presence of the light band ruled out dispersive replication, since it would have resulted only in intermediate blended bacterial DNA. So Meselson and Stahl's experiment supports the semi-conservative DNA replication model. Here are my sources. All graphics on this presentation were created by me using Inkscape, a free software for illustrating. Thanks for watching.